All right, hello. My name is Craig Roderick, and I will be your lab instructor for the fall 2020 section for 1147. This is my phone number, 937-859-7292. You can get me early in the day from 7 to 3:30 at my email, craig.roderick@beavercreek.k12.oh.us. All right, that is the best way to get a hold of me early. I can check eLearn and my school, Sinclair.edu, does not always work, so this one is a really good one. I can check it once in a while. Sometimes it pops up, sometimes it doesn't. Don't know what to say about that. But let's get in. I'm going to try to record these labs, which will give you extra stuff to learn. The objectives for each lab are there. That should be your direction. That will tell you what you need to know. I'll try to let you know what to emphasize and what to kind of ignore to save your time. So I'm going to do these labs video-wise as if I was doing these here, which I am. There's just nobody here except you're watching a video. The cool thing probably about this is I can get these done really quick, and I don't have to worry about people getting behind, because you can stop the video, roll it back, and redo it and listen and get your notes in that way. What I try to do with each one of these labs is try to pick the part of the lab that would take you the longest amount of time to do. The rest of the lab is usually just grunt work. It's just looking up, indexing, writing down, that sort of thing. And I'll let you know when we get to those different labs. So some labs I do a lot, some labs I do a little bit, and some labs I don't do a whole lot. Those are the ones I actually like the most. But I probably do really make sure that students know this material in the lab. It's supposed to be a student-centered lab. Maybe I would do that different if I worked here during the day, but I know that evenings we have the demographic is so different than in the daytime, and what you're working with is people that have jobs, have kids, have spouses, and the thing that the instructor should do or try to do is reduce the amount of time as much as possible that it takes you to do this material. So over 18 years I've been able to figure out exactly what to give you that puts all the information in your fingers. I don't know how to answer certain questions relating to remote learning. This is new, it's new to everybody. So if you ask me a question, I'm going to probably say I have to get back with you on that. All right? We're just going to learn this as we go along, as we did it a little bit last semester. We just kind of rode with it, and you know, it ended up being pretty good. Students did very, very well in my lecture. They did very well in my lab, and I don't see any reason why that's going to change. So the material I'm going to give you is through this lab that I'm doing. I kind of want to make sure the thing is still rolling. Yeah, it's still going pretty good. Hopefully I'm centered where I need to be. And so we did a good job, and so you're going to have these videos as if you were still here. There are other things I'm going to download to you in terms of PowerPoints and pictures and things like that. There's already a shell that has been moved that you'll be able to see in your content that has already been placed there. To be frank with you, I haven't even looked at it because it probably just got loaded last night. I actually had to have one of the full-time professors do it for me. So I made her a student in our class, a member of the class, and then as a co-instructor, and she was able to transfer that stuff over because we haven't frankly used this stuff. I'm going to work on a couple of students. I'll probably call you to see if we can set up a what's called a virtual lab meeting. It's in eLearn under Tools. And so I'll try to walk you guys through that. So just a lot of stuff to get done. So I really want to make sure I get these videos done, plus the normal content that I give you anyways, to help you with each one of these labs. So if you get in your lab book, if you turn to page 3, our first lab is the microscope. Basically, what lab is, or A&P is all just basically structure and function. Can you identify a structure, and can you identify a function with that structure? And so lab is definitely a place where we're just looking at structure and function. You're only going to get asked 2 questions. A piece of tape is going to point to something, and they're going to say, what is this? And you'll have a multiple choice, and you have to tell us what it is. Or I might point to something, and I'll say, at the pointer, what does that do? What is its function? So there's only 2 questions to ask in lab. What is it? And what's its function? So not very difficult. Some people might find it difficult, so maybe that was a wrong thing to say. But a lot of people, and you've all done difficult things before, so this won't be any different. Hopefully I reduce the difficulty. Go through your objectives, all right? There are 4 of those, and you'll notice on page 4, that is the meat of what we're going to be doing. If you were to label these off to the left on the microscope part, you'd see that there are 13 parts to the microscope, and there are 13 functions. Sometimes there's multiple functions, so make sure you know both functions. That's only fair, all right? For instance, this is called the eyepiece, but it's also called the ocular lens, as you see at the top of page 4. So what I'm going to do is I'm going to go through the 13 different parts, and we're going to talk about stuff that's on page 7 and page 8, okay? Page 5 and 6 are just diagrams of this microscope right here, and you actually have a real live microscope in front of you. So let me make sure that my microscope is in focus. I want to make sure that it's back far enough. I might get my zoom off of this. Right there. Yeah, the whole time I was talking, you probably couldn't see me, but you know what? I'm doing this by myself, so there is no director, no cameraman. I am it, Jack. This is what you have right here. So let me kind of focus that guy right there in the middle. There we go. Okay, I'll stand to the left. Actually, I'm having to do this over again because I had the audio messed up. So we're going to go through this again, and I'm going to go much faster. All right, eyepieces right here. You have 2 of them. This is a stereo microscope, also known as ocular lens. Take a look at what the function is. I'm going to try to save my voice. Take a look at that. Lens is what you look through view, and each one gives you a 10-time magnification. So if you were to look at a specimen that was on a slide right here, you would see it 10 times larger than life. This is the arm right here, and the arm supports the body and stage and provides a handle for carrying. 2 functions. Make sure you know them both. The revolving nose piece. This is this guy right here. And what does a revolving nose piece do? It holds 3 different lenses called objective lenses. Each of these objective lenses has a different power. The weakest one we call the scanning. It is only 4 times. So in other words, if you look through here, you'll see the specimen 4 times larger than it is in real life. But you have 10 times in your ocular lens. So 10 times 4, 40. Yep. If you take a look at page 8 at the bottom, Roman numeral 4, how to calculate total magnification, we just did that, and it's written there for you. All right, so we'll be able to complete low power and high power. Just put 10 times, okay, for the ocular. Just fill those two spaces in, and we're going to fill in the next lens. The next lens is low power, which is 10 time magnification. 10 times 10 is 100. Maximum magnification, high power, is a 40 times lens times 10 gives you 400 magnification. So if you're looking at a specimen, you're now seeing it 400 times larger than normal, and that allows us to see most tissues of the body in pretty good detail. Now, one of the things that you have to understand is that there is a trade-off for increasing your magnification. Why do we use a scanning objective lens? Well, sometimes we want to be able to see the entire field of view in which we have tissue on this slide right here. And in order to know where in that slide do I want to focus my attention, we use a scanning microscope. So if you look at page 7, if I had the letter E and I want to know all about the letter E, by using my lowest power magnification, scanning, all right, that is 40 times larger, you can see the letter E, but you can still see everything around it too if there were other things around the letter E. We called that the field of view. So at low magnification, you have your greatest amount of field of view. If I increase magnification, all right, I increase my resolution, I increase my magnification, but I lose field of view. So as you can see, I see all of the letter E on page 7 in the low power field, and I've lost some of the field of view that I had when I was using scanning. Now, if I use the highest power magnification, all right, I can't even get the whole letter E into my field of view, but I have my greatest magnification. So what we're trying to show you here is that if you increase magnification, you lose field of view, all right? If you increase field of view, you lose magnification. That is the dichotomy that you need to understand. In lab, and I don't know how they're going to do this, but they're going to do it somehow, they might have 3 microscopes, A, B, C, and they'll have each one set up differently. And so one might set up with the scanning, and they may say, which one of these microscope has the greatest or least field of view? And the one that has the least field of view would be microscope A, right here, okay? Or that would have the most field of view, sorry, least amount of magnification. Or they may have one set up like this. They have it on high magnification, and if the question is which one has the least field of view, then you would pick this microscope as it is set up on high power, all right? Okay. Stage is this square black thing right here, okay? Let me see if that's in focus. Yeah, close enough. Get it right there. Yeah. So the stage is what holds, see, gravity keeps it from going down, and I put it in this little silver thing with an arm right here, and this is my mechanical stage. So you got the stage and the mechanical stage holding that, okay? And make sure you look at the functions for both of those, all right? If I want to move my mechanical stage forward and backward, I use what is called the mechanical stage control knobs. They're located underneath the stage on the right-hand side of the microscope, all right? Once I get my specimen centered where I want it, I then use my scanning, get my stage all the way up, and I use the coarse adjustment knob to move the stage down until it gets kind of in focus. Then I stop. Then I move to the fine knob right here. So if you look right here at the bottom of the arm near the base, you have a huge knob right here, and as you can see, I can move the stage halfway down with only a quarter turn. And when I go to the fine knob, the fine adjustment is used only when I have high power. So for the coarse adjustment and fine adjustment, make sure that you understand when to use those. All right. The condenser. The condenser is a way to control light in coming from the lamp. The lamp is in the base right here. And I usually have students put their finger like this and then move it up, and you hit something called the condenser. The condenser is this guy right here. You'll notice that there is a filter in there. Okay, it's purple. And purple, we use what's called irosine red dye. And we can see different shades of pink, purple, and red with this. If I take that out and put this back, then I won't be able to see anything. Okay, but let me take the filter out because I want to show you this right here. You'll notice that in the condenser you have this diaphragm that can open and close. So lots of light in, very little light, lots, little. Okay, and we need to be able to control the amount of light because let's say you're looking at tissue that has cilia, these little hairs that line the apical side of the membrane of the cell. Okay, apical means top part, basal means underneath. And so these little hairs, okay, if you had tons of light, the light would just your retinas would pick up all of that light and there'd be no blocking the light, and so you bleach the retina and you won't see anything. Okay, so if all of a sudden I want to see those cilia, I need to get as little light as possible, and then once that light is nice and dim, you can see the light is blocking. That's why they call this a contrast microscope compound, and I can basically then see those fine things. So the condenser concentrates light. The condenser height knob is over here on the left side. Okay, it's right here. You can see that. And that just moves the condenser up and down. The idea is to bring the condenser all the way up, back it off a hair. That way, if you focus too much with this right here, these stages don't allow it, but the old microscopes, you used to be able to drive one of the objective lens in through the specimen slide and crack the condenser itself. Okay, the iris diaphragm is this wheel right here, and that's what adjusts the light. The illuminator is right here. That's the light bulb. And the light switch is over here on the side, and a rheostat to control and dim the light. Okay, so two ways to control the light: iris diaphragm and the rheostat right there. All right, those are your 13 parts of the microscope. Okay, detailed on page 5 for you, close up on page 6. Page 7 was about field of view and magnification. Roman numeral 4, how to calculate the total magnification for this microscope. All right, so that is the microscope lab, and next video will be on molecules.